Morning, everyone. Thank you very much for being here. And I'm going to go over culture versus next generation sequencing and what are the differences. Diagnosis of periprosthetic joint infection, PJI, is currently a very challenging problem. As you know, there is no absolute test. Hence, clinicians who encounter a suspected PJI case have to use a combination of tests, all of which, besides their expense, can be invasive, and even they are far from perfect. In 2013, the MSIs came up with a set of criteria for defining PJI, which was then modified by the International Consensus Meeting. And that definition also got modified in 2018 and now has a scoring system that includes a preoperative and intraoperative phase. However, all these criteria are heavily based on culture because culture is part of the major and also part of the minor diagnostic criteria. And as Emanuele mentioned, culture negative PJIs are on the rise. And in the literature shows that up to 34% of PJI cases, that's more than 1 out of 3 patients, are having negative cultures. So it's a huge issue for us that can compromise diagnosis and timely management of these patients. Culture is the traditional method of detecting infection and has been around for more than 200 years. And it's interesting because I cannot think of any other technology that has been around for this long without a major change. There are several disadvantages that are inherent to culture. One is the yield of culture is as low as 30%. And in order to battle this problem, we oftentimes obtain multiple samples to be able to increase the yield. Cultures are timely and they are costly because, again, we're taking multiple samples and to run for a single patient, that just adds to the cost. And one of the other problems that oftentimes gets missed is that cultures take physical space. And I know at Cooper, our microbiology lab always complains about all the samples that we send, and we want to keep them for a long time for slow-growing organisms. And that's a challenge that and a burden that is put on the microbiology labs. The other problem with cultures is that there is a lot of lab variation, and they are technique dependent, and different culture mediums can yield different results. And oftentimes, the results of culture can also be confusing because let's say you send 4 samples for one patient and only one of them comes back positive on a random medium like thigh broth. It is extremely difficult to make a conclusion out of a situation like this because now you have 1 out of 4 that is growing something, so you cannot ignore it. At the same time, you have 3 negative results. So now whether this is contamination, is it false, like all those negatives are false negatives, these are all imposed challenges on a diagnosis that is already a challenging process. In order to unify the way the cultures are made and the cultures are processed and kind of synchronizing all the labs, the Infectious Disease Society of America and American Society for Microbiology came up with a set of guidelines. The main idea is that for cultures, you need to have a live microorganism so that it can grow. And looking at these guidelines, you can quickly see that we are not really following them because, for example, aerobic and anaerobic samples need to be transferred into a room temperature container immediately. And I'm sure that you have oftentimes seen the picture that I have in the bottom left, that the cultures are sitting in these boxes outside the clinic for almost 24 hours in 100 degrees in a summer day. So that just essentially kills the bacteria, so you cannot have a live organism to grow. There are several limitations that are mostly specific to orthopedics and to periprosthetic joint infection. One is the issue of biofilm formation. Again, for cultures, you need to have a live organism. to be able to grow something out of it. And with synovial fluid aspirations, when biofilm is formed, the yield for capturing a live organism is extremely low. The other thing is that the time to diagnosis is extremely important for management of these patients. Oftentimes, you need to wait 14 to 21 days for cultures to conclude. And if you want to do a DARE procedure, you're going to lose that golden window just waiting for your culture results to come back. So now what's the solution? I think next generation sequencing can significantly help us with that. And in a nutshell, with next generation sequencing, we're matching the DNA and RNA molecules that are captured from a specimen and match them with a database that we have with the mapping of all the genomic information of these organisms. Just a little bit of story and history of NGS. It all started with the Human Genome Project in 1985. That was a study that was sponsored by NIH, Department of Energy, and Department of Defense with $3 billion at the time. That was the highest sponsored study of all times. And they tried to look at the genome and the map of the human genome. And almost 20 countries contributed to the study and collaborated on it. It took them about 10 years to have the complete mapping of human genome. And at a time, the cost was around $2 million, and it would take them 1 year to process and have the complete mapping. So as you can tell, it was not really a practical solution. But I want to grab your attention to the graph that I have on the right side of the slide. As you can see, in early 2000s, there was a huge drop in the cost, and that's where the next generation sequencing was introduced. And that's why now we have companies like 23andMe or Ancestry that kind of provide this test at costs below $100 just for the fun of it to see what your genomic mapping is. And I want to grab your attention to this slide, and that's essentially what next generation sequencing does for detecting infection. On the left is what culture is. You get a seed, and you want to make sure that that seed is alive. You plant it, wait for it to grow, and based on how it grows and the pattern of the leaves, you can tell me that it's maple. Or the alternative is what we have on the right side, have an atlas of all the pictures of all the seeds, and as soon as you pick it up, regardless of whether or not it's alive, you can match it with a picture of another seed that is in your genomic database and tell me right off the bat that it is maple, for example. And I think that's how NGS can significantly reduce the time and accuracy for diagnosis. Again, like Dr. Parvizi said, I highly encourage
Thank you, Alisina. Much appreciated. I know I echo, but don't worry, I will stop. Next, we're going to have Professor Smith. He's a professor of microbiology. Mike did an amazing job last Saturday, and we're looking forward to learning from him again today.